So thank you so much for giving us the opportunity to present the French Gut Initiative to this audience. We are all aware that some environmental factors can be risk factors for non-communicable disease. We also know that some of those factors might modulate the gut microbiome. Thus, the question is whether the gut microbiome is a mediator between environmental factors and non-communicable disease. The microbiome then can be seen as a target to develop preventive or therapeutic approaches, or a source of predictive or diagnostic biomarkers. And that's to answer this question that we launched the French Gut, le microbiote français. So the French Gut aims to recruit 100,000 participants by 2029, healthy and sick people. And there is a cross-sectional aspect to the project. We will collect metagenomic data, 20 million reads, health, lifestyle, and diet data through self-reported questionnaires. And there is also a longitudinal aspect to the study, since we are connected to the National Health Data System that will give us the medical history retrospectively 10 years before and prospectively 20 years after the time of recruitment. So altogether, this data will help us to meet the scientific goals that are threefold: define the gut microbiome reference of the healthy population, pave the way for precision and preventive nutrition and medicine, discover early biomarkers of disease and potential targets to treat the disease. So we wanted to take the opportunity of this presentation to present you some of the challenges of this project and present you how we believe we overcome some of the challenges. One of them is how we recruit 100,000 individuals. For that, we use heavy communication through different media, traditional media like TV, radio, podcast, press, but also social media. We have today more than 15 ambassadors, influencers that help to recruit actively participants through the social media like Instagram, TikTok, Facebook, and YouTube. And you can see here the recruitment curve, and we have today more than 25 participants and biobank samples.
Another challenge with the French Gut was to upgrade all the processes we had at Metagenopolis to improve the number of samples we were able to process in a year, which before the French Gut was about 3,000 samples to now about 30,000 samples that we can process in a year. For that, we had to test different steps from the process, from the aliquoting to the DNA extraction and purification and sequencing. And for that, we used different mock samples, testing before and after all the optimization to see if we were able to provide microbiome profiling, the same level of the theoretical composition from mock samples, or testing also real samples, DNA fragment generation quality. And for that, we were able to generate and optimize this automation pipeline with this huge biobanking, followed with the aliquoting, DNA extraction, and purification, metagenomic preparation, library preparation, and sequencing. At the same time, at Metagenopolis, we also have bioinformaticians that are expert in analyzing microbiome data, and they developed a new software called Meteor 2, which provides a combined taxonomic, functional, and strain level profiling. And this software got the accuracy of the comparable other softwares that exist today, but also has the capacity to detect very low abundant species that are difficult to detect in some specific cases that could sometimes participate in the disease. And this also detection of low coverage species allows for a better detection from possible cross-contamination during lab preparation, with cross-contamination that could come even at very low rates. And this helped us to really improve all the processing of upgrade we have in the French Gut. With the French gut, we also opened the opportunity for ancillary studies with different partners for which are running different cohorts, but with a highly phenotype analysis. And we will provide for them the human microbial fecal sample composition expertise. The French gut is also a combination of multidisciplinary consortium, which bring public institution and private institution that help us to generate this high quality, large scale study. The French gut is not just a French national study, it's also part of all the large initiative in the world, with the French gut being part of the Million Microbiome Project. And with that international effort, we help each other, providing new standards for improving the processing of the microbiome analysis, and try to create this one of the largest human microbiome database with the horizon 2029. And in 2025, we will open the French gut not only to adults, but also to the kids, for adults from 3 to 17 years old with the French gut kids. And also open analysis to overseas cohorts, like the cohort that we have with La Réunion in collaboration with INSERM and colleagues at Université de La Réunion. And for that, we thank you very much for this opportunity for presenting the French gut. We would like to finish by thanking all our colleagues at INRAE and PHP and all the French gut and Million Microbiome Consortium that help us creating this amazing project and making it available for the community. Thank you very much.
Thank you so much.